Well, hello, everyone. Welcome again to our developer community meeting. It's February 21st, and today we'll have another guest presentation. Before I jump in and let Arvind start his presentation, I wanted to just quickly go over a couple community updates. So just as usual, if there is a meeting topic that you're interested in having Cerebras discuss at one of these meetings, please email us at developer@cerebras.net. Additionally, if you haven't already, please join our Cerebras discourse page. There's a lot of good conversations that are happening there, and discourse.cerebras.net is the link to join. Recently, we launched an update to our AI Model Studio, which is our cloud product. This is the ability to now fine-tune large language models from the cloud. So we have some models that are available today: GPT-J, GPT-NeoX, CodeGen, and more are coming soon. So if people that are interested in fine-tuning via the cloud through Cerebras, please share our website with them. With that said, I'll hand over the sharing responsibilities to Arvind. Arvind's from Argonne National Lab, and he'll be presenting his team's research today.
So thank you, Uday. So my name is Arvind. I'm a staff scientist in Argonne National Lab, and this represents a fairly large collaborative effort which has been going on for a while now, about a year or so. And we'll be talking about how to build language models for genomic language or genome-scale data sets. And this is kind of an interesting question because when we think of genomic data, we normally don't think of language models as the first source of, algorithm to analyze them. But I'm sure you've heard of this from a variety of sources, including Google and other places where they've done quite a bit of work with these transformer networks, large transformer networks for these sorts of data sets. So I'm going to talk a little bit about how we built genome scale language models and how we adapted them in order to understand the evolutionary dynamics of SARS-CoV-2. And, there's fairly a large team that was involved in actually doing this work. We were, surprisingly, we actually won the Gordon Bell Award for some of the largest science runs that were set up as part of this story. And the Gordon Bell, if you have not heard of it, it's one of the technical awards given for supercomputing applications. It's considered like the Oscars for supercomputing. But it was a fun experience to actually share this and try and do this on very large data that we had access to as part of the story. And of course, we were collaborating with Cerebras for a while even before that. And the story is actually an evolving one, as I'm going to talk a little bit about. All right. So why is this an interesting question to even look at? So if you want to understand how a variant actually emerges, it's actually a very cumbersome process, unfortunately. So we all know that the virus, once it attaches itself to the human body, can get inside the cell and start replicating itself. The virus replicates in many different mechanisms. One of the ways it does that is actually having a copy machine inside of the cell. And what people have to normally end up doing is track all these variants that are coming out of these patients at any given time, and then laboriously organize them into these phylogenetic trees. And this is actually kind of, a very high fidelity virus in the sense that it acquires about 2 mutations per month. And the reason for that is because it has a complex proofreading mechanisms. And only some mutations in the viral genome can give rise to these new phenotypes that cause increased transmissibility or severity and so on. And the variants with increased transmissibility and severity are the ones that we usually call as variants of concern, because these are the ones that you need to be worried about. And this usually takes anywhere between 4 to 6 months for people to check out these variants of concern, because there's quite a bit of analysis that needs to be done. And currently only Omicron remains on the variant of concern today, because that's the one that's fast spreading. But other variants in the past include Alpha, Beta, and Delta. Right? But if you were to take 6 months before we even identify a variant of concern, that's actually a problem, because it's not going to be something that we can wait for Otherwise the disease would spread, and actually you could have quite a bit of a handful, which has been seen throughout the pandemic so far. So therefore, there is a need to develop these scalable or automated reliable tools that can identify VOCs and sort of maintain this continuous watch list of variants that we should be tracking. And only then we can actually basically say if there is something interesting happening. The variants of concern actually emerge from very complex interactions between the whole viral genome and the host. So if you think of the work that's been done for a very long time, until recently for understanding the viral evolution, it's been mostly focused on this particular protein called the spike, which is the first protein that basically binds itself to the human cell, right? And within the spike protein, there is a receptor binding domain, which is about 300 residues or so, which is a very small portion of this 1,000 residue, domain that's expressed by the virus. And if you look at the number of mutations, obviously that is the one that you're like mostly tracking, but that's not very useful, because if you really know how many interactions happen between the host and the pathogen, it includes the spike. It also includes the nuclear capsid, which is one of the most abundant protein in the virus itself. And this affects infectivity, transmissibility, and severity. There's also the membrane protein and the envelope proteins, where there are some mutations that are important to track. And there are some non-structural proteins also, which enable them to acquire new mutations. And that's kind of important for us to track. And the whole genome scale evolutionary models will eventually provide us with this insight of what are the potentially vulnerable regions within the virus. And if so, we can create this continuous watch list that I was talking about before, and you can understand how these evolutionary strains impact the creation of new variants that we need to tackle. And that's also like providing downstream information for like building new vaccines. So that's the biological background between why we want to actually track at the whole genome scale. The basic hypothesis that we went with in the study was to basically say, is it possible for us to adapt these large language models to describe how the SARS-CoV-2 genome actually evolves, right? And why are we doing this at the level of the DNA instead of at the level of the protein? Is that the DNA actually has quite a bit of information regarding all the translational modifications that can happen even before it's actually translated into this protein. So thus far, a lot of people, including people from Facebook, other companies which have been building these language models, they have mostly relied on protein alphabets, and they have had great success. And these parameters, the number of parameters in each of these models actually is quite impressive. They go all the way from 670 million to about 50 billion, which is awesome in terms of, how much of data they can actually look at and what is the sequence length that they can do. The GenSlim models, the genome scale language models that we are building, builds on success of some of these approaches like DNA-BERT or the N-formal model, which still have fewer number of parameters, but they were very much trained on very limited data sets. On the other hand, we actually had access to over 110 million gene sequences, which is where the foundation models were actually trained on. And then we refer to the models that were fine-tuned on the SARS-CoV-2 as GenSlim models itself, which were actually a fairly small number of parameters, 50 million or so. And the data set size was about 1.5 million sequences that we had seen for the SARS-CoV-2. And then you have the GenSlim diffusion model, which I'll also present a little bit about in terms of understanding how you can expand the length of the sequence, in order to get the correct context. And that's actually a model that has about 2.5 billion parameters. And that was used to train on these, SARS-CoV-2 sequences. So the reason for choosing DNA over all other representation is that there is this inbuilt hierarchy that we already understand. A single gene is approximately about, order of 1,000 base pairs. Viral genomes are between 10 and 100 kilobase pairs. And then you have plasmids and pathways, which are like between 10 and 100 kilobase pairs. And then you can go all the way to human, which is about 3 gigabase pairs. So, there is this neat hierarchy of interesting events that happen at each scale of the genome. And then we wanted to basically say, can we build models that can actually scale to an entire genome, right? So on the first hand, this paper is one of the first ones to report a genome scale large language model for understanding SARS-CoV-2 evolution. And in fact, one of the things that actually surprised us was the fact that the 3 papers that were actually selected for the last year's Gordon Bell competition, all of them were using language models. Ours was the only one that was actually building on genome-scale language models. And in terms of the algorithmic advances, what we are planning to show is that the language models that we built for genome scale representations actually learn very meaningful latent representation. So that includes GC content and understanding how sequence lengths and other evolution incur certain costs for these proteins to or genes to actually work. Then you have the codon level representation, which we claim has more biological information than the 20 amino acids or 20 natural amino acids that we can look at. And when we start training these models, one of the quick things that we realized was that because there is not much diversity in the sequences of SARS-CoV-2, we needed to actually build what are called as foundation models first. And these foundation models were trained on these raw nucleotide sequences from about 25 million to 25 billion trainable parameters, using more than 110 million gene sequences, which accounted for sufficient diversity in the data. So how do you do this? You have to scale these foundation model trainings somehow. We use GPU-based supercomputers because that's actually bread and butter in some sense for many of your supercomputing community. Basically, Selene at NVIDIA gave us access to their systems, which allowed us to train it on 4096 GPUs. And then you had Polaris, which is our Argonne supercomputing cluster, which has about 2048 GPUs. And then we were using the Cerebras CS2 wafer-scale engine cluster to enhance how we could train the entire model on just several CS2s that are built together as a cluster. So in terms of the algorithmic advances, we also needed to worry about the sequence length because 10,240 codons or 30,000 nucleotide sequences is not a small length in any context. So most natural language applications usually focus on shorter templates or shorter padding sequences, usually between 512 to 1024. Here we went all the way to like 10,240 or 30,000, depending on how you're using the sequence itself. And that's actually a fairly long sequence on which capturing the context is actually important. So we have to develop some algorithmic advances that include this idea called as hierarchical diffusion. I'll give you an idea of what that is. And then finally, we are also integrating this into an end-to-end workflow that allows us to characterize both the genomic and biophysical data to identify these variants of concern, right? And this whole application took us about 850 petaflops of mixed precision peak runs, which is kind of cool to see how these large language models can actually, chew on computers. So one way to think about how we do this is to basically say you can train on the genome sequences that are directly available for SARS-CoV-2, but that is unfortunately not such a great idea because less than 1 to 2% of the entire genome actually changes. So that's why we said it's possible for us to think about this in the case of a foundation model, where you can pre-train these models on 110 million sequences that come from this very large database called as PATRIC, or Bacterial and Viral Resource Center, which maintains all the gene sequences that we know of. And then you can fine-tune the model on the SARS-CoV-2 open reading frames. Open reading frame is basically a collection of these genes that are put together. And then you can periodically retrain on new variants when they come across, and then using a specified time window. And then we are going to measure performances on CS2, Selene, and Polaris. So these trained foundation models are the basis for how we are thinking about, how we train and build these models. And from the trained foundation models, you can actually extract what is called as a semantic similarity score, which basically tells you, given a particular variant, how far away it is from the average of seeing all these sequences. And there's also the sequence log likelihood score that basically lets you interpret how likely is this particular sequence to occur given all the things that we have seen. So these two correspond to two important aspects from a biological perspective, which happens to quantify whether the sequence can escape immune detection. So that basically goes into this component of immune escape score. And the second one happens to be the fitness evaluation, which basically tells you if the sequence is more likely to be seen, then it has a better fitness. And so you basically assign a better score for that. So, and then you basically combine these two scores to become what is called as a variant of concern score from the language models. On the other side, you can also have these diffusion models that let us predict the next set of genes or next set of evolutionary variants that you're going to observe. And that diffusion model can basically have complete SARS-CoV-2 genomes, which you can still embed them into the latent space and observe what is the variation looking like. And then you feed them back into tools such as OpenFold, which is AlphaFold's open version. And you can measure the sort of epitope alteration and the protein-protein interaction scores using MD simulations. I'm going to talk about this anyway in greater detail as we go along, but these are the things that constitute this entire workflow, starting from the foundation model all the way to the place where we actually are like able to detect if this is a new variant of concern or not. Right. So with that, let's get into the first portion, which is to say, how did we build our foundation model? So the foundation models are actually what are called as genome-scale language models. I already spoke a little bit about this. It's nothing but a simple causal language model that's implemented, very similar to GPT-2. And the input sequence is what you basically take as the input, and you have a set of transformer layers that have attention also with them. And then you create a semantic embedding from that. And once you get the semantic embedding, the input tokens are basically codons or groups of three, letters that are stringed together from the nucleotide sequence. And then it basically retains the semantic embedding that tells you what that looks like. And then you can translate that into the protein sequence world once you actually get those scores. So the GenSlim Foundation model looks at a sequence length of 2048, which happens to be the maximum length of all the genes that we have looked at so far. And it varies from all the way from 25 million to 25 billion. And then you have the other models that I spoke about, which were trained on the SARS-CoV-2 genomes, which is also like this, same number of parameters. But the sequence set is much smaller because it comes from a smaller set of sequences that we selected for the SARS-CoV-2 genome. The scaling part of it is actually the biggest concern. It takes O of L squared, where L is basically the length of the sequence that you're looking at. And you need to overcome certain communication overheads and parameters and checkpointing, which need to be like clearly one of the bottlenecks for training such large models, especially when it comes to like the 25 billion model. And, this represents one of the largest foundation models that were trained on the raw nucleotide sequence themselves. When it comes to like looking at qualities of the training itself, we looked at aspects such as this perplexity. Perplexity is basically a measure that tells you, how many guesses are required by the large language model to predict one token of interest. Basically, if you're asking for, if you mask a token and ask it for the prediction, does it do it really well? And the perplexity of 1 usually implies that it's a perfect model. And what I want you to pay attention to here is just don't pay attention to the 25 billion model right now, but if you pay attention to the 25 million, to the 250 million, and the 2.5 billion, you can sort of see that the perplexity of the model improves quite a bit as you actually increase the number of parameters. For the 25 billion model alone, model sharding and training times were actually imposing a lot of constraints on the GPUs in terms of training them. So that was one of the reasons we actually said, maybe the Cerebras CS2 Wafer-Scale Cluster is the way to go for this, because if you want to train such large models out of the box, it's going to be a very hard thing to do on just the GPUs. Right. So once we had that aspect, we were able to like look at these models and say, we're going to choose the 2.5 billion model because that has the lowest perplexity of all the sequences that we did. And in terms of the infrastructure for training the GenSlim models, we basically used Perlmutter, Polaris, and NVIDIA. And as you can see, Kyle has basically pasted himself on all the 3 machines so that he can basically say he was on these 3 systems or system architectures as part of the training. But we also used the Hugging Face interface for all of our training runs using the transformer libraries as well as the PyTorch Lightning and Deep Speed for accelerating our training algorithms. The model itself is fairly simple to use. So if you basically say, if you import the GenSlim model directly, you can basically instantiate these models. You can input a sequence and it'll basically tell you what that embedding looks like, and you get an output from that. So it's a very simple, straightforward application of GPT-2-like architectures. But the idea that we could do it with genomic scale data, and then we could use this with the models that we already had built, is kind of cool. And we actually get meaningful results out of this. In terms of scaling, right? So what I want you to observe here is this is now showing what happens with respect to each of the models with the number of parameters. These are foundation models that were scaled in Polaris and Selene. And you can see that we've seen almost linear scaling with the number of parameters that we see. At the end of the day, the larger models are the ones that actually show better scaling with respect to the communication overlap. And then, the performance of the deep speed scaling actually drops as we scale to like 512 GPUs. And that can be avoided if you end up using, DDP directly from PyTorch if you want. And that is also something that we have tried, but we didn't present the results in the paper because, this is kind of interesting from what deep speed can really do. For the smaller model, Selene, which has 80 GB per node in its GPUs, achieves 2x the times of throughput than what we see from Polaris, and therefore you're able to double the batch size. That also allows for better training and stability over the course of the runs. And one of the things that we also observe is performance runs in Selene with 25 billion models at 4,096 GPUs can achieve about 121 petaflops of sustained runs and about 850 petaflops of mixed precision peak, runs. And, the total number of flops that it takes for us to run this, it's actually like quite large. It's about 1.60 zettaflops total. So that was kind of cool for us to see that a large language model could take up so much of computing time and effort in terms of like running on the system. The foundation models, though, are quite powerful. And I'm going to talk a little bit about the biological insights that we get from looking at this data set. So what you see here is a TSNE embedding of a small subset of the 110 million sequences that we took over. And each dot here is actually a gene. And now I'm going to use that to kind of illustrate some of the properties that we learned from the data. So every gene is now painted with the sequence length of that particular gene in terms of the number of codons. And what I want you to observe is this neat division between things that are like high and low sequence lengths. And you also see the formation of several islands that have like high sequence length numbers versus some of them that are like low sequence numbers, and some of them which are like the average lengths that you observe throughout the landscape. So this is kind of interesting to see from the level of organization. Now, the other thing that you might also want to do is to paint this with what is called as GC content. So the GC content is basically a way of saying, how does a gene adapt itself to new environments? So if you've not heard about GC content, it's a measure of the stability of the G and C pairing in your DNA. And that is usually increased or decreased depending on where the organism is actually like working. So GC content usually is a illustration of, how much adaptation has happened throughout evolution for these genes to be at certain locations. So one of the things that is kind of interesting to note is that there is a very nice gradation of how the GC content is varying. And depending on where that organism probably came from, you could basically say this is coming from a certain location or not, just by looking at the GC content. So which is kind of cool for us. And the fact that we could cluster the data so well also led us to question something about, if there are related genes that have similar functions, how do they lie? So here is an example of this protein called as malate dehydrogenase, or the gene called as malate dehydrogenase, which encodes this protein called malate dehydrogenase. And this malate dehydrogenase enzyme, as you can see, there's one region where you have like high GC content versus another region with low GC content. And when you actually start looking at those structures, we actually get them to be in two different isoforms. And they're clustered separately just because of that reason, because they have like completely different isoforms, but the same function. And they also belong to some, roughly different groups in the system. So that was kind of cool for us to expose certain functional aspects of these organization which could not be directly, evidence from normal ways of analysis. So this is kind of the, money shot for saying, why do you want to use genome scale models in the first place? So now that I've spoken a little bit about the, how we use this for training our, foundation models, let's talk a little bit about how we train them on the SARS-CoV-2 open reading frames. So one of the things that we wanted to challenge the model was basically to say, is it possible for this model to see only 1 year's worth of data and then try to predict all of the rest of mutations or all of the rest of the variants that has been seen after that? So what was interesting was we basically said we're going to limit ourselves to give only the first year's data from the start of the pandemic. So it basically has seen very few of the other variants thus far. And then he said, is it possible for us to train the model on that and then see if it can distinguish the variants? So again, the money shot picture, what does happen is the fact that we can use GenSlims to kind of understand the differences in the variants. And, And it also tells you that with respect to the different alignment scores, we do see them to be clustered somewhat differently. And why do these actually cluster this way is also because there is some biogeography involved in actually like setting those strains to be from the same, strain. So it also classifies in some sense the country from which these sequences came from. And you can actually clearly see that we can sort of identify alpha, beta, gamma, and delta, lambda, omicron sequences very clearly from this set of sequences. And these islands are mostly formed by the country of origin from which they came from. We don't know all the regions that are actually changing as a consequence of geography, but we can definitely point out the locations where they're mixed well. For example, in this bottom area where you can see them to be more or less mixed well. And also here where there are some regions where you can see this sort of like clustering happening with respect to the different strains. And now I can ask this question, what are the differences that you see from the model and how do you interpret that for the differences? So we sort of created this visual to kind of help us easily understand what was going on. So what you see in the circular plot is this entire open reading frames of SARS-CoV-2. And from the SARS-CoV-2 open reading frames, we can basically identify the proteins that are expressed by these open reading frames. It includes NSP 1, 2, 3, 4, all the way to 16. And then you have the spike and the other proteins that are expressed as part of the membrane and envelope proteins. In the internal ring, we actually paint it with the sequence log likelihood that interprets the attention values from the large language models. And then you also have the evolutionary coupling that happens because of the attention parameters that basically tell you how one region of the genome is coupled evolutionarily to another. So in this case, this happens to be the iota sequence where you can see that this log likelihood is quite small all over the place. That's because you probably saw very few sequences of this type in the data. And naturally, that's a region which has like low log sequence likelihood. On the other hand, when you take a sequence that is coming from gamma, I think, or actually delta, I'm sorry, this is a delta sequence, you can sort of see that the coupling is actually quite different. You can see a lot of the sequence having high regions of confidence in terms of the overall areas where you see the sequence log likelihood to be higher. And then you can also start comparing them with respect to the different regions that are coupled to the spike protein, for example. So if I take the spike as the highlight, you can sort of see how tightly the other sequences are coupled to the different regions versus how it is loosely coupled. For example, you can see this region does not have any coverage in terms of the co-evolutionary patterns. But on the other hand, you see some increased interactions between the other proteins. So these sorts of evolutionary couplings, which is kind of impactful for sort of identifying how these might be mutations that we need to be paying attention for new variants and so on, comes into the play. Now, in terms of training these genome sequences on the entire SARS-CoV-2, it was not possible to do this on GPU-based clusters because we were not doing any model parallelism. It is possible to train them, but it would have taken us a very long time. It is at this point we decided to use the Cerebras Wafer-Scale Cluster. And I'm thankful to Cerebras for actually saying that this is possible. And at that time, we said, okay, let's take the GP2 backbone, 123 million to 1.3 billion parameters, and use the entire sequence length of the SARS-CoV-2 genomes as our input. And it uses this memory-X and swarm-X architecture, which lets us basically, distribute the weights and gradients in an intelligent manner so that you can do the input pre-processing and then run it on the entire cluster of CS2s that are connected. So in terms of the money shot again, this does provide a significant reduction in the time to solution. So if you really think about the number of training steps that we needed to reach convergence, so this is a busy slide, I apologize for that, but with one CS2s, it takes about 4.1 hours to train. With 4 CS2s, we can actually reduce the training to about 2.4 hours with a smaller model, 123 million parameter model. On the other hand, the GenStem with about 1.3 billion takes less than 10.4 hours to reach convergence, which is kind of cool because you do get perplexity values that are like close to ideal, like 1.025. And this is also reflected in the fact that we do see these embeddings that are nicely clustered with respect to the rest of the sequences. And this density of plot basically represents all the codons, all the sequences that are colored by the sequence length. And when you increase the number of devices, as you see on the left-hand side, you also see as you increase the number of parameters, the time to solution actually significantly goes down. So with a larger number of, devices, you're able to accommodate a much larger sample size, and that lets us train it with, within less than half a day for the entire genome sequences. So this is sort of this idea that you could take these models and, without much change to your code, you can run them at scale on the CS2s, which really let us get to this point where we could train the entire model on a cluster of CS2s without any trouble. Of course, if you ask Cindy and others that were part of the Cerebras team, they could give you all the gory details of what went internally. But for us, what was even cool was the fact that we could train these very large models without much effort on our end, right? And you can also see that they cluster very nicely with respect to the different sequence types that we were looking at, including alpha, omicron, iota, eta, lambda, and delta. And that's kind of nice to see the sort of clustering that you see, similar to what we saw before, where you also had the clustering with respect to the biogeography of where these sequences came from. So this is kind of a very nice way to induce how we could use Cerebras in terms of training much of these larger models. Now, coming to this idea of how do we train and get these diffusion models to work? So the diffusion model as such, as we know, the computation grows quadratically when we have to compute the attention values on much larger sequences. So the typical solution is to cut these sequences into smaller chunks and learn them. But the issue would still remain, which is that it doesn't capture the long-term sequences or long-term dependencies that exist. So the idea was to introduce this high-level module that lets us capture these longer-term dependencies using the diffusion approach, which is kind of this neat idea that you could use for basically distinguishing something that is real versus unreal, and then use that to basically denoise the data and get more insights into how we train those models. So we use the pre-trained foundation model, which is a 2.5 billion parameter one, and then use the diffusion model in terms of learning these longer term contexts. And here is an illustration of how that works. So we batch these sequences initially with initial, single codon space or single nucleotide space, and then it tries to predict what happens when you group them into like 5, 10, and 20, codons at the same time. So the hierarchical diffusion model can generate these SARS-CoV-2 genomes that maintain the context, which is kind of important because if you look at the number of open reading frames from the original data, you can see the distribution. It's always between 7 and 9, which is what you want to keep it at. But the baseline model, without any hierarchical diffusion, if you just do the baseline generation model, that ends up generating a lot of these invalid sequences. So the model does definitely include a better set of representations if you use the hierarchical diffusion. So once you basically get these scores, we can basically assign variants of concern and things like that, which is what I was speaking about when we lost connection. But the generative models are kind of powerful. So, you can start generating new sequences. Here is the gray sequences which are coming from the data that was collected versus the red lines which are coming from the generated sequences. And what I want you to pay attention to are particularly the red lines, because if you look at the number of sequences that it is generating, it's mostly proportional to the variance that we have seen so far from the rest of the tree. And just remember, when I told you in the beginning, these trees were actually the data for the training was actually coming from only the first year of sequences. We never used the other sequences for this generation process. And it's able to generate these sequences that look like Omicron or Delta and so on with a certain confidence, and we can say how much that looks like by actually comparing them with sequence characteristics that we use. So for example, we can basically say, is this close to some of the neighbors that we have seen so far? If so, then this is not a variant that we need to be worried about. On the other hand, you can also see if these variants are like close to other things that we have not seen before, which means that these are like truly, maybe these are new variants that we need to be worried about. And that's actually the portion that actually helped us, make the story in some sense, because we could generate these synthetic sequences that look like SARS-CoV-2, but they also had the same number of open reading frames, that also had the same quality control that we could actually perform and look at them in terms of their similarity to other sequences. So what was even more interesting at the time, this was in December or November when we presented it. At that time, there was this new variant called as BQ1 that everybody was talking about. And the BQ1 variant is kind of this thing that basically could bind to the ACE2 receptor in much stronger sense. And one of the sequences that we actually generated had better binding to the ACE2 receptor, which is what is shown by these MD simulation calculations, where you can see we can compare these generated variants. So GB top P213 or 123 and so on are the generated variants. And we are basically comparing them to alpha, beta, gamma, delta, and omicron sequences to kind of see how far or how close they are in terms of the binding. So what happens is one of those variants that we did end up generating, which is GB top P123, so far seems to have the best quality in terms of binding to the ACE2 receptor that actually has a number of mutations that are all spread over the entire protein. And you can see the mutations that are highlighted. And those are the ones that form tight interactions with the ACE2 receptor. So we were kind of happy to see that the variants are not nonsense, but they're actually meaningful and they can be modeled and they can be used to basically quantify whether we need to pay attention to those sequences or not. So that basically summarizes the entire story for how we have been developing our workflow, including these biophysical aspects of like simulating the binding, score calculations and epitope alterations and so on. But, it's one of the largest large language models built for genomic data from raw nucleotide sequences. It achieves near linear scaling with the CS2 wafer-scale engine cluster, and basically allowing you to scale the solution in such a way that you can reach model convergence within half a day, which is almost unheard of with large language models. And then you have this novel integrated workflow that allows you to visualize and analyze the data as you're like looking at it. This has a lot of implications just beyond SARS-CoV-2 also, because it provides you with functional data into the organization of genes. Because it's learning codon bias and also trying to model evolution, we are able to capture a lot of these environmental adaptation features. What I didn't speak about was, wet lab validation of some of the experiments that we did in terms of generating new enzymes and things like that, where we have been also successful in looking at them in terms of validating them in the lab. And there are several applications beyond COVID-19, which also includes the fact that you can now annotate proteins for different functions, which you might not have an idea what they actually stand for. You can also do a lot of vaccine design and sort of test them in vivo using these approaches. And then there is a complete distributed workflow that enables us to characterize variants of concerns on whole genomes. It basically integrates a number of AI approaches together. So it basically says you can chain large language models with structural biology AI, such as OpenFold. And then you can also integrate this with fast neural operators, which I didn't speak about in any detail, but that's joint work with Animal and Kumar at NVIDIA. And then we can also do these for genome scale inference with biophysical methods, which has actually been a challenge for the community. Of course, this work wouldn't have been possible without a whole bunch of people from NVIDIA and Harvard. Claire, as well as Natalia and Cindy, were instrumental from Cerebras in getting these models stood up and run and tested. I know they spent a lot of time, so we are eternally grateful for them actually having spent a lot of time doing this. And of course, my team from Argonne, a whole lot of people that were involved in like design and working through the day-to-day algorithms and how we could test them in the workflows and so on. And then, Joe, Victor, Janet, Silvio, and Daphne all worked on this visualization piece, which is kind of critical to communicate our story. And of course, Rafael and Lee and Lucas were basically essential for getting us data, Globus access, so that we could store these very large data sets. Just to give you a sense, the embeddings themselves run into several petabytes. So it's not a small data set by any means, but it's kind of this thing that we want to make it open and everybody should be able to access it. And if you have any interest in looking at the code is freely available. You're also able to access the model weights as well as the data directly on the website. And the funding was made possible with the DOE National Virtual Biotechnology Lab, as well as the Candle workflows. And with that, I'd like to thank you for your attention. And if you have questions, please go ahead.
Yeah, thank you so much for your presentation, Arvind. This was a really insightful presentation. I'm going to ask a quick question, and then obviously others can jump in as they wish.
Sure.
So you presented a lot of this research, and it all tails to COVID. But I'm assuming that this work can be leveraged to accelerate research into the genomes of other bacteria or viruses. Would you be able to speak a little bit about what that technique would look like? Is it just fine tuning from the foundation model, or could you leverage the fine-tuned model itself?
Yeah, great question. So there are a couple of things to pay attention to here. One aspect is the fact that when you're looking at these language models, these were first trained on these gene sequences which have a reduced sequence length. And so when you're now chaining together genes in a specific order and putting them in a line, they do have this issue where you have to worry about the context of saying, does it cross the length of the sequences that you're looking at? And that becomes a problem when it crosses a certain length limit. So say beyond 10,000 nucleotides or 10,000 codons, it's probably not going to do very well. And that's a space where we are trying to explore these hierarchical diffusion algorithms and trying to see how we can improve on them. And we are now trying to do this with E. coli genomes, which represent one of the smaller bacterial genomes on which you can work on. So the proof of the pudding will probably come in the next couple of months, where we will know whether we are able to use these language models directly on E. coli data. And if it's able to basically do simple predictions in the beginning, and then see if we can adapt them for much longer genome scale sequences. But the idea is to basically go there. I mean, that's exactly why we even played with these models in the first place, and that's, I think, where we'll go.
Thank you.
Excellent talk. Thank you. Maybe a related model question. Is there any way to kind of simplify, or maybe just focusing on the CS2 pattern, so kind of make something like that more accessible to people with fewer resources than what was brought to bear in the garden bed experiment? Because it strikes me that at some point we want to get this to a point where this is actually available to more people in their investigations, be it to do with future pandemics or just a general research environment. So SC and Gordon Bell always goes for heroic stuff, but then maybe it's then also an interesting question if one can kind of downscale to address, for the use of lesser resource people?
Sure. Yeah, I think that's a great question and also required by the community. The way we have envisaged this project, at least for now, is that all of the models that were trained on both Cerebras and our GPU-based clusters, all of the models and the weights and everything is available. You can download it directly. Just be prepared for an onslaught of data. That's the only thing I would say. And you can use the models directly for inference and things like that. So that's not something that we want to keep it close to ourselves or anything. So everything is available online, and we are free to download it from the paper and the website I pointed to. So if you go to my lab's website, you should be able to download these models easily. So in terms of like scaling down the parameters, we actually built these models in such a way that you could train them with like 25 million or 250 million, even on regular GPU clusters with about 4 to 8 GPU nodes, right? If you have a DGX box, for example, you should be able to compile and run. So that's another possibility where we have provided templates for these models. So you should be able to like really change them and run in any way that you'd like. So if you don't mind, this is not for like self-promotion in any way. Just that my students did a lot of work in making sure that they could, we could get these codes to actually work the way that you want. So let's see. Do you see my screen that is shared with the browser?
Yeah, we do.
Okay, good. So these models, if you look at it, the way we have developed is basically provided you with templates to say, okay, if it is 25 million parameters, you can run it on a single GPU, and how would you do that, and so on. So everything is available in such a way that you can do these models in different examples are also there. So you can do the training examples where we have the foundation model, which is all set up in very easy to use YAML format. So say you want to do this on the 25 million model parameter, we have all the different sets of things that you need to run. And we do give you the correct config that you need to use and so on for the models itself. So it's pretty nicely stored and nicely configured in such a way that you don't have to kill yourself in terms of setting it up and running. And this reflects on what others have done with the Hugging Face models also. So if you have the Hugging Face interface, you can directly work with it. So that's not a problem at all, right? So, and they also have a command line tools to make it easy for you to deploy to your own supercomputers or your own systems internally. So that's also something that we have been working to make sure that others will have access to it, right? So.
Thank you very much. That's excellent.
Hey, Arvind, I'll ask another question.
Sure.
You mentioned in your presentation that you used 1 year's worth of COVID research or COVID data.
Mm-hmm.
If you were to, I don't know, continuously fine-tune your model, would you expect the performance? I'm assuming the answer is yes, but would you expect the performance of the model to drastically improve, or would you just see nominal gains?
No, I think it does improve quite a bit. The reason we challenged ourselves with just looking at the 1 year's worth of data was to see what is the extent that this model can really push. Right? So, and that's a minimal amount of data that we could say with some confidence that it has some sequences to look at that are interesting. And it then went on to do what it did. So that was like a different story. As we continue to improve the models with different sequences or different diversity levels, it does produce better results. We haven't done rounds of fine-tuning on the SARS-CoV-2 genomes yet, but we will be doing them shortly because we do need to publish that paper where people are asking us, oh, why haven't you done this or not? And then we are like, we first need to finish writing this one paper with the foundation model, and then following that, we'll have the SARS-CoV-2 paper. which also talks about this. So that's the plan of action for now. But at that point, I think we'll use all of the years worth of data and show how much the model actually increases its performance, or does it degrade, or does it become very specific to just the branches that it has seen. So we'll have more ideas then.
Great. Well, if no other takers, I'll continue to ask questions since I have quite a few. You also, I think on one of your previous slides, you showed that perplexity reaches near zero. I think it was probably closer to 1.025. And I think that's using probably the biggest size model, or the largest size model with the largest sized sequence length. What would you say, I guess it's a two-part question, what do you think had the bigger influence, or had a substantial influence on reducing perplexity? Would that be the ability to train with larger or longer sequence lengths? And then if you had, infinite time and resources, what would be maybe the next thing that you would think about in order to further reduce perplexity?
So walking that aspect of optimizing it for perplexity is a double-edged sword, right? So you don't want a model that's near perfect. Otherwise, you basically say you're memorizing the entire data, which is not what you want to do, right? So it has to have some predictive power. So in order to make sure that we have predictive power, we end up choosing models that have more simpler number of parameters. And we go back to the original data and ask these questions of what level of variation you see in the data itself. And this actually comes through empirical experimentation. It doesn't come I don't think there is a very clear theory behind how to set up these parameters for your runs or for your application per se. From what we have seen so far, we seem to believe that increasing the model sizes definitely improves the perplexity of the model, and perhaps even also have better predictability in terms of some complex tasks, right? So some of these things are being tested right now on the foundation model, where we get to see a little bit more about what does it mean to have higher perplexity versus models that have the balance of perplexity versus model sizes are smaller. But I, for one, prefer models that are smaller but have higher predictive value, right? Than having sequences that you are able to reproduce the sequence very correctly, but if it doesn't have the biological significance covered very well, then there's no use in using that model. So that's something that we'll have to play and see in the way these models are being trained. So far, I think this experiment has been done only with the shorter sequences, where we know that the models have better perplexity when you go through this one. And with the Cerebras model, when we were training them too, right? A perplexity of 1.025 with a model size where you only see 2% change in the number of sequences that is in the sequence itself. Only 2.5% out of 10,000, or maybe it's 45 to 50 sequence positions are changed, and that it's able to predict it with great accuracy should not be surprising, especially on Cerebras. My hope is that with the foundation model, we'll see some more behaviors that might help us advance this behavior, advance this a little bit more. So we'll have to see what happens when you train the foundation model on the Cerebras cards, and if they actually end up getting better performance or better perplexity. So we'll see.
Awesome. Thank you.
Yeah, great presentation. I've got a quick question.
Sure.
Hi, so my name is JP, and I noticed that when you were looking at the config file, that the epoch was, I think the batch, you had 8 epochs total. And
yes.
I wanted to take a look at that, and then also understand from an optimizer selection standpoint, and then also how that impacted your learning rate.
Oh, great question. So epochs is a huge question when it comes to these large language models. When usually you observe these large language models, the way it is that they probably don't go more than a couple of epochs on the text corpora that we have been playing with, right? And 8 epochs here, we don't even actually like cross more than a couple of epochs in order to reach convergence. So I would say that that's mostly 8 was put there for, keeping it sane for us. Like we'll be more comfortable when we have a larger number of epochs. But from the point of view of how many steps we did train, it was less than an epoch, really speaking, because even with even with 500, 600 iterations, in some cases about 3,500 iterations, where it had roughly seen 25 to 35% of the data, the models all converged pretty much nicely. So there's not much of an improvement in terms of the model performance after that. So we basically held off in terms of like saying, okay, let's not push it to a complete epoch that might not be really necessary. And this is true with most large language models. People don't train for a whole lot of time. In terms of the training optimizer as such, the optimizers, I think we tried a bunch of things. I wish Alex or Max were here. Max has left the group now to find greener pastures. But Max basically tried a bunch of different experiments where he said, I'm going to keep this learning rate and then try this optimizer, soft warm-ups and things like that. And then what he found that worked for him was this add-in optimizer that had this soft way of like reducing the learning rate over the course of the iterations and then speeding it up a little bit later. So that soft warm-up really helped him to do better training at the end of the day, is what I would say from what I understood from his experiments. I think in the model config, I think we do say what optimizers we use at the end of the day. I think in the Cerebras one, probably Natalia or Cindy can answer it better than me, because we were not involved in that decision of saying, do we have to run it using a particular type of optimizer? They basically try to keep it as close as possible to what we had done. So that's one thing to just keep in mind. There might be some differences in the way Cerebras chose to optimize versus what we chose to optimize on the GPUs.
Yeah, no, I think that's great. I have a follow-up question. I think you kind of read my mind there in terms of like looking at the hardware when it comes to the optimization, because I know that one of the benefits with Cerebras is that with such a large amount of like, cores per wafer, you can really kind of keep as much of like the map moles on like chip itself, right? Instead of having to write them. So then I'd love to like hear kind of like that thought process of like, oh, we have this, advanced wafer. How can we kind of optimize for its use case instead of just, kind of more general GPUs?
Yeah. So that's a great question. And for which I think the answer partially depends on two things. One is how large is your large GPU cluster? Right. So really speaking, what happened in our case was because we didn't have at the starting point of the project itself, we didn't have a very good handle on what we would consider a Cerebras chip equivalent on our GPU cluster. Right. So we said, okay, let's assume that the DGX cluster is the best that we could do, which has 8 GPUs attached to it. And that's where we'll start. And if we are able to beat that with the Cerebras, of course, then it would make sense for us to even consider having the Cerebras as a contender for this sort of a job. And of course, with working with engineers at Cerebras, we quickly realized that that sort of a comparison again is not apples to apples, because there is a lot of things that go into considering how you set up your model on Cerebras and what goes into getting the best bang for the buck in some sense. And so once they showed us how to do these tricks, or how we learned to do these tricks from them, it really helped us basically finesse the message for saying, we have this very large problem. Can you help us, get to a point where we can solve this? And that's when I think Cindy and Claire and others basically jumped in and said, oh, this is actually something that would be right up our alley, because we have also been thinking of very large sequence lengths and so on. And we particularly restricted ourselves in terms of using the large sequence length as our challenge problem, which in retrospect might not have been the best way to think about it, but I think I'm quite happy with the way it turned out when we had to run this for the runs that we did. But yes, for us, the equivalent was basically a poor man's DGX in some sense, and then try to say how that would actually compare and contrast with the sort of performance you could get with the Cerebras. And then that was a genesis for making decisions eventually of how this would look like on a full cluster.
Wow. Okay, no, very, very insightful and very, very interesting. Thank you again.
Thanks.
Oh, and thanks for Uday Modi. I don't know if I'm pronouncing your name correctly, but thank you for setting this up. This is great to have the community get together and discuss these topics.
Yeah, no problem. And all thanks to Arvind for taking the time to provide this presentation today. I know we're a little over time, but if there are any last questions, yeah, please use this time to ask them now. All right, Arvind, thank you so much for presenting today. We really, really appreciate your time and your research. We look forward to seeing what you and your team do next. And yeah, to all those who joined today, thank you also for attending.
Thank you so much. Appreciate it. See you.
Awesome. Thanks all.
Bye.
Bye.
Bye.